So for those of you who don't know, what is a mooneye? So it is a long, silvery fish, very flat body profile, about 10 to 14 inches long, and the tells that separate them from other fish that look similar to this are the large eye that takes up a significant portion of their head, and these bony, tooth-like structures on their tongue. So the population of Mooneye has been decreasing over the last couple of decades in New York. But the Oswegatchie River seems to be holding the best known population that we have, which makes it prime for doing this study. And there's very little research on the actual New York populations of Mooneye. So that's kind of where we come in. So what we do know about this fish is that they do not tolerate turbid waters very well. They're a clear water fish. They prefer solid, more rocky substrates as opposed to sandy or silty. And that they can be reliably caught in 3 to 4 sites in the Oswegatchie River and then 1 site of the Indian River, which connects to the Oswegatchie. And in past experiments with this, their catchability has been pretty reasonable in the spring portion, and then summer to fall, it kind of dips off and then rebounds just slightly in the fall. So our objectives coming into this were to establish an accurate population and distribution estimate, to characterize the habitat and why they're using this certain parts of the river certain times of the year, to identify the fish community surrounding Mooneye, so what other fish might be present in the same areas, and to get a little better handle on their actual spawning behaviors, and hopefully putting all of this together to draft a conservation plan for Mooneye in New York. So the detection methods that we have been using are electrofishing, gill netting, and a bit of citizen science. The electrofishing seems to be more safe for the fish. There's a much lower mortality rate, where with gill netting it can be up to about 40%. But the gill netting is much easier to implement in certain parts of the river, where the electrofishing is an 18-foot boat. It's a pretty heavy boat, so you need a pretty substantial ramp to be able to use it. Where gill netting, you've got a 14-foot john boat, you can just kind of slide it in wherever you need to. And a bit of the citizen science. We're just trying to get in touch with anglers, get them to report their incidental catches to us, ideally with a location and date that they caught it, and preferably with a photo so that we can confirm that it is a mooneye. So the electrofishing, we've been able to get out about 3 days a week, which is pretty good for trying to get 3 or 4 college student schedules to line up to do field work. Trying to keep our distribution effort about the same across our sites, and sampling each site for 1 hour to try to standardize our protocol. For the gill netting, we are using 150-foot long gill nets, floating gill nets, 6 feet tall, that are trisected into 3 mesh sizes, 2, 2.5, and 3-inch mesh sizes.
Can I stop you, Dave, to explain how that is very specialized, being almost at the surface?
The floating gill nets? Just that the top line is a very buoyant line, so it just keeps it, I guess, floating near the surface. So it samples just the top 6 feet or so. And then what we were trying to do was sample right around dusk, about 3 hours a night, about an hour before sunset to 2 hours after. But in the instances when we did actually catch mooneye in the gill nets were times when we actually let the nets sit overnight, recommended by Mr. Carlson. But the trick with these is that, per the FEMRIF proposal, we're not supposed to gill net when the water temperature gets above 23 degrees C. It's just added stress to the fish, the mortality rate goes even higher. So we're a little bit limited in the summer when we can actually gill net. For the citizen science portion, we've been distributing flyers like these around the Oswegatchie River Basin, just trying to drum up that attention toward the fish and get the attention of the anglers who might be able to tell us where they've happened to catch these fish. So just to give you an idea of our sites, this is the Oswegatchie River right here, a bit of the Indian River. And the three sites that I mentioned before are Flat Rocks, Hubleton, and Eelweir. Then on the Indian River we have Rossi, which you can kind of see the start of Black Lake here. This site would be way down in here on the other side of the lake. These are the sites where most of the catches have occurred, so we focused on these sites to try to establish our population estimate. Then right around mid-July, we tried a new site just to see if we could expand our distribution a little bit. And we actually found a couple Mooneye up near Ogdensburg, so we were able to add that to our distribution, started throwing that into our rotation of sites. So for the habitat characteristics, we're collecting standard air, water temperatures, the turbidity, conductivity for the electrofishing. And wherever we did detect Mooneye, we would try to get into the stream flow to see if they're in sort of the faster headwaters of a site versus the slower tailwaters. And about halfway through the summer, we purchased a fish cam. So when we were able to find where Mooneye were, we could actually drop the camera down and see what the sediment looked like in that area, what the vegetation looked like. So far, from what we've seen, it's very flat, not a lot of vegetation. So this is kind of a summary of our efforts for the season. The electrofishing just seemed to have a higher catch per unit effort, Mooneye per hour, versus the gill netting. And throughout the season, we were able to deploy 18 tags. We had 33 observations, including some angler reports. There's one in there that the fish was just too small to actually tag, so we had to only count that as the observation, we couldn't tag it. But not too bad for having 64 site visits for the season. A lot of our observations were kind of front-loaded in the spring. We started off really strong, only 2 sampling days in April, where we came across 6 fish. May, we caught almost a quarter of our fish for the year. But then, once you got into June, it really started to fall off. July is a little bit deceiving here because of the 4 fish, 3 of those were an angler report by the same person. They caught 3 fish in one night. We only handled 1 ourselves. So the fishing really fell off for the summer and then just started to recover. This was like the last week of October, and then had a few fish in the very beginning of November. So kind of plotted that against the water temperature. As the water warmed up, it correlated pretty nicely with the fishing going down. As the water warms up, what fish tend to do, go deeper, try to get that cooler water. So we kind of thought maybe we weren't detecting them just because they're getting out of range of the electrofishing boat. The range of that is only about 10 feet or so. If they're getting deeper than that, it would be hard for us to detect them, or if we are affecting them, to actually see that we're affecting them. So kind of saw at the beginning of the year, we were catching them in 7 to 9 feet of water, and then there was a pretty hard cutoff around June, where we didn't see any fish in less than 10 feet of water. So it's a possibility that they could just be too deep for us to detect with the electrofishing boat. So as far as our population estimate, we're just trying a mark recapture style experiment. As I mentioned before, we were able to deploy the 18 pit tags, and we only had the 1 recapture, which unfortunately wasn't even one of our pit tags that we deployed. It was just this small guy right here. We thought he was too small to put a tag in at the time. We saw he had some injuries on him, but that is actually how we identified him, was these abrasions on his back. When we caught him again, we were able to look back at photographs we had and see that it was the same fish. So the conclusions we were able to come to after our first season was that they do like that fast, cold water, and they're easily catchable in the springtime. But after early to mid-June, they definitely moved. We were not catching them in those areas. So we kind of need to figure out where they're going from there. and that we probably need to start adjusting our survey methods since we weren't catching them late fall. We just need to try something a little bit different this coming season. So going into next season, we want to try to expand that citizen science approach a little bit more, get more flyers out, maybe not just in the Oswegatchie watershed, but maybe in the Racket and Grass as well, just try to get a bigger pool of fishermen. More aggressive sampling in the spring, try to get around that spawning time a little bit more. Last season, I was still going through a lot of the training to be able to operate the fishing boat. I had teaching assistant responsibilities as well, so we kind of missed a lot of that prime time. So this season, I wanted to be able to maybe get ahead of that spawn a little bit. And maybe shifting the electrofishing toward a more dusk time, rather than trying to do it late afternoon. Maybe there's a little bit more chance that they would be coming to the surface to feed. So try maybe shifting that time a little bit, see if that produces better results. And trying to utilize the gill netting a little bit more, just to try to change up that sampling style a little bit. And we focused a lot on the population estimate this year, so we hit those same 4 or 5 sites repeatedly. It would be nice to be able to get out and kind of broaden our horizons a little bit, try some new sites and see if we can detect some Mooneye. So special thanks to my advisor, Dr. Langen from Clarkson University, Fish and Wildlife Service, and Scott Schluter for all the equipment that they have let us use. Endless amounts of information from Mr. Carlson and Glenn Johnson, FEMRA for the funding, and New York State DEC for their cooperation. Of course, everybody who set foot on the research boat. It would not be possible without that.